Let's move on, folks, to talk about tertiary and quaternary structure. So tertiary structure is the three-dimensional structure of the entire protein and all of its side chains. The quaternary structure is a spatial arrangement of more than one individual protein, and we'll call those individual proteins subunits. Back to the slide real quick. So tertiary structure, we can see that we've zoomed out from looking at secondary structure to see what is the shape of the whole protein. And then quaternary structure, you can see, is when more than one individual protein comes together. And in this picture, you can see this protein has 4 different proteins that come together to make its quaternary structure. Let's talk a little bit about tertiary structure. So oftentimes in tertiary structure, we'll take a look at how the alpha helices or the beta sheets come together, and then we'll give that structure a name. So the units of secondary structure combine to form some type of motif that is common among proteins or a domain. Disulfide bonds can help to strengthen tertiary structure. Remember, disulfide bonds happen between 2 cysteine residues between their sulfur atoms. We have kind of 2 main types of tertiary structure. The first is fibrous, like keratin, like collagen. We're really thinking like long fibers is the tertiary structure of these types of proteins. The other types of proteins are called globular. This is like everything else, right? They're just kind of globs, and we can further define what the glob looks like. Some example of globular proteins, we have a helix bundle. I'm showing you 2 different cartoons here, folks. So alpha helices in a helix bundle, shown to you as this bundle of ribbons, or in a 2-dimensional cartoon, you'll oftentimes see the alpha helix as like a cylinder. In the middle is a type of tertiary structure called an immunoglobulin fold. We do not have to be memorizing these types of tertiary structure, but just recognize, hey, this is an entire protein, it's showing me its tertiary structure. You'll see this is comprised of beta sheets because we see these ribbons with little arrowheads on them. In a 2-dimensional structure, again, we see those ribbons with arrowheads. And then finally on the farthest right, we have a beta-alpha beta fold. So this contains both alpha helices and beta sheets. You can see alpha helices, you can see beta sheets in both the 3-dimensional and 2-dimensional representation of this protein. Now, of course, in all of these proteins, you can see they contain turns and loops, right? Because turns and loops are what is connecting the alpha helices and the beta sheets. So here's some turns and loops. Here's some turns and loops. Here's some turns and loops. Every protein is going to have turns and loops. Now, because we have proteins that can have very similar types of tertiary structure, we can have domains. Particularly, proteins that are large are going to have domains in them. A domain is a distinct region within the same protein. So it's a distinct region within an individual protein. In particular, for proteins pretty big, it's going to have some distinct regions within it. If a protein has more than one domain, more than one region, we expect those domains to have different functions. Maybe one of them is a signaling part, maybe one of them is a chemistry part. And each domain will be comprised of kind of multiple layers of secondary structure on top of each other to create this tertiary structure, to seal off a hydrophobic core. Save that in your mind for a second. We'll come right back to it in the next video. To wrap up domains, folks, domains are recognized as structurally similar. Any domain will have a distinct function, and in this class, you'll learn, oftentimes, that structure equals function. So if this domain has a very similar tertiary structure to this other domain on a different protein, if the structure is the same, we'd assume the function is probably the same as well. And an example of that is on the slide here. So for example, cytochrome C has a domain that is very close, very similar in structure to many other types of proteins. Look on the slide below. You can see that all of these 4 proteins shown have this kind of red thing in the center, and they're surrounded by these bits of peptide that are all very close to the same shape. So even though all of these proteins are distinct from one another, they are different proteins from different organisms. Since the structure is the same, they have the same domain, we should assume the function is the same as well. In the next video, we'll talk more about tertiary structure, protein folding, and the hydrophobic effect.